I'm Ayaan, and I'm 13 years old, and for the past few months, I've been researching into pharmacogenomics, which is the study of how your genes affect your response to drugs, and I was recently able to personalize blood clot medications to specific genotypes, and I'm also working on a nanotechnology project with Samarth, in which we're using nanopores to sequence genes much more effectively than companies out there today. Samarth himself being a machine learning developer and a blockchain developer. And with our knowledge, both in genetics, we decided to be able to find a cure to all viral infections, including cancer and diseases. And this is our mission at Revive. Right now, we're tackling the problem with antibiotics and radiation therapy, but it just doesn't work. Firstly, because different bacteria have different PBPs and they have different natures. For example, cancer cells have certain receptors which don't allow all drugs to work on them. Secondly, because certain drugs, such as chemotherapy drugs, cause side effects, and they also harm good bacteria, which is not what we want, and we need this bacteria to be able to do functions in our body. And lastly, because the medication isn't personalized. And so if we're giving somebody a medication that has a high toxicity, and we're not giving them the right dosage that's personalized to their metabolizer, they will die due to high toxicity of the drug inside their body. Our vision at Revive is that by 2028, the mortality rate due to these viral infections will go down by about 70%. Our process works in 3 main steps: radiation screening, a lysine protein, and drug delivery. So first, radiation screening. Now, usually when you go to the doctor, you'll use radiation screening to screen all the cells in your body from head to toe. And this is what we'll be doing to be able to figure out where the infected cells are in your body. So we'll come up with an image kind of like what we see on the screen, and we'll be able to tell where the cells are because they'll be highlighted in white. But now that we know where these infected cells are, how exactly are we going to be able to kill them? Well, this is where our lysine protein comes in. Every virus has DNA, and these virus microbes have certain variants that code for certain proteins, and these proteins allow for the cell to be able to harm other bacteria which we need in our body. But technically, if we take out these coding instructions in the first place, the fundamental DNA that the cell needs to live will be left with a blank cell, and so therefore the cell will no longer exist. This is what we aim to do with our lysine protein. We'll be using a technique called phage display to be able to create this protein. Now, it's a technique in which a gene encoding a protein is inserted into a gene, and this is all inserted into a phage. So our protein will obviously be the lysine, and the gene encoding the protein will be specific to the area that we want our drug to go to, or the exact function of our drug. So now that we know how to kill the infected cells and how to find them, how exactly is this all going to be implemented into the body? We'll be doing this through a drug delivery process. We've created our own drug, which has 2 drug compartments, in which we have 3 exit ports. Now these exit ports are used for the phages to come out of their compartments and out of the drug in total. We have 2 compartments which have both of our phages inside of them, and these phages hold the lysine protein. And lastly, we have our push compartment, which has a water storage inside of it. Now once this water storage is filled, it'll create osmotic pressure, and it will release and allow the exit ports to break, thereby allowing the phages to also go into the area of infected cells. Now, this isn't just some cute sci-fi idea. We've actually talked to professionals in the field. For example, Ruslan Dorfman, head of CEO at PillCheck, and Salima Baidani, a pharmacist at Whole Health Pharmacy, and some other people from U of T as well. At Revive, we have enough data to be able to solve this problem, and we know that we will. Our vision is to reduce the mortality rate, and we want you to join us to make this into a reality. Thank you.